Since 1977, the Ceiba Lions Club has been a pillar of service and community care on the island. What began with 20 committed members has grown into a legacy of giving back, supporting those in need, and advancing the global mission of Lions Club International. From cultural events to signature service projects like Meals on Wheels and the Food Bank, the Ceiba Lions Club supports the elderly, disabled, and struggling families with compassion and consistency. Their health programs, like annual eye care support and glucose fairs, continue to promote wellness across the island. The club is also a champion for the environment, proudly maintaining an adopted hiking trail and backing local cleanup efforts to protect Ceiba's natural beauty. Several of its members have been honored with the prestigious Melvin Jones Fellow Award for their outstanding dedication, proof that the spirit of service is alive and well on Ceiba. The Ceiba Lions Club, serving with heart for the good of all.
Good evening, everyone. Thank you. Thank you, everyone, for coming, both on Ceiba and to our live stream audience. Our audience is a little smaller tonight as we are a little concerned about Tropical Storm Jerry out there, but now that we've prepared and gone inside, for sure there won't be a drop of rain. So, again, thank you for coming as always. I'm going to find my clicker. Sorry, a little off here today. And go back. As we know, those of you who have been watching or been coming, the live streaming events that we are doing can be seen live or after the fact. They are on Facebook for maybe 30 days, but they're also on YouTube, so you can look at previous presenters as well. And that's made possible by the Lions Club. Our events are not possible without multiple sponsorship. So these are our key funders. So CCG is the Caribbean Culture Fund, formerly Prince Bernhard Culture Funds, Public Entity Ceiba, and the Nature Environmental Policy Plan. So these are key financial funders. Of course, this month-long project is not possible without a lot of support, including more than 50 sponsors. So we can't mention all of them every night, but by all means, have a look at those logos, and when you frequent any of those businesses or properties or see these people, it's super helpful to us if you thank them for making this possible. Our speaker tonight is generously sponsored by the Cottage Club. They have been a long-time sponsor. Beautiful property right in Windward Side. The other thing we do is we do a raffle every year. This year, we're specifically targeting our raffle, and our goal is to sell 1,000 tickets. So that would raise $5,000. It's only $5 a ticket. And we are specifically trying to fund our scientists January through March when we run Critter Quest. And Critter Quest is specifically to establish the baseline data of our invertebrates on the island. Very important to monitoring climate change, and a very efficient, low-cost way of doing a monitoring method. So by all means, support the raffle. And you don't have to be present on Ceiba. The drawing will be October 27th. You can even watch it on live stream. Upcoming events. Hopefully, no weather issues. But right now, we are still planning our 8 o'clock in the morning hike on Saturday with tonight's presenter. And we are confident that once you hear her talk, you will want to be out there in the amazing world of fungi. You can sign up tonight, contact us another way, and we got a whole booming weekend of events. So at 3 o'clock on Saturday, Mikhail Volkin, who's in our audience tonight, is doing his official field project to check out, find more insects. On Friday, the slide says that we're premiering David Attenborough's, they tell us it's his last movie, and it's a wonderful premiere. You can't see it anywhere else, but you could see it on Saba if you're lucky to be here. But because of the weather forecast for tomorrow, we preemptively moved it to Sunday at the Cove Bay. It is free of charge, but it's also meant to be a nice evening. Wear blue in the spirit of the ocean and watch an incredible movie for free, or stay for dinner as a full nice evening. There's a nice dinner menu as well. And that's Sunday, is it, Emma? Oh, sorry. Upcoming event, sorry. On Saturday, Joe Wonderly is doing our next amazing presentation, and that's back at Amanhana. I'm one step ahead because I'm thinking Sunday. On Sunday, we also have a tropic bird hike with Mikhail and Joe, and that morning, we also have a bird watching event that I'm doing. Not me, but with Joe Wonderly as well. So all kinds of stuff to do this weekend. We're going to get rid of this bad weather tomorrow and then be on our way for a great weekend of activities. If you do a field project with us, you get one of our cool hats, a bucket hat or a baseball hat. They're made from recycled plastic. They're really cool, and just another reason to join us. So here we go with tonight's presentation and our introduction. Prepare to dig deep into the fascinating world of mushrooms with Dr. Jean Lodge. For over 26 years, Jean has studied the hidden fungi that keep tropical Caribbean forests thriving, from speeding up leaf decomposition to preventing soil erosion after hurricanes. Her work reveals just how vital these often overlooked organisms are to forest health and resilience. Now an adjunct professor and curator of the Georgia Museum of Natural History, Fungarium, Jean continues to discover new mushroom species and inspire students with her passion for forest fungal ecology. She's truly a fungi girl to be around. Please join me in giving a well-deserved round of applause for Dr. Jean Lodge.
Thank you. Thank you for the very nice introduction. I was actually working in Puerto Rico for 36 years. But yeah, you saw the warning. There is sex and some violence, so be prepared, be forewarned. This is a three.
You said that ahead of time, there'd be more people.
Oh. This is a three-part. How mushrooms slow erosion in wet tropical forests and how climate change affects that. That's important for where we were in Puerto Rico. It's important for high wet islands like Saba. And then I'm going to talk about my favorite group of mushrooms, the wax cap fungi, and how they evolved in the Caribbean. And then, because we're doing the run-up for Halloween, you get special fungal stories for Halloween. Okay. Now, how many of you know that fungi are closer to animals than to plants? Most of you. Okay. Fungi have, most of them, a linear structure. They're made out of hyphae, except for the yeast, which we like for the bread and the beer and the wine and all that. So these linear structures grouped together, they can be hyphal fans, like you see here and here. They can have cords and hyphal strands, like you see here, connecting two leaves. or they can have rhizomorphs, and that literally means root-like. And if you can see in here what looks like horse's hair, and this is in fact the horse's hair marasmius. And it grows up in bushes and down on the ground. Birds like them. They are very handy for tying their nests together. They repel water, so they keep the eggs and the nestlings drier than your regular organic material. And what my colleagues in Tennessee learned from culturing the rhizomorphs that I was sending them, that nothing else would grow in an agar plate with them. No bacteria, no yeast, no fungi, nothing. These things have really powerful antibiotics that are unknown. And they may be helping the birds with preventing disease and destruction of eggs and diseases in the nesting. You can see that the fungi are tying these leaves together. And that's important, and that's what I'm going to focus on the first part. That binding those leaves together into a mat makes it like throwing a rug out on the forest floor, and it really doesn't move much with the pounding rain and even overland flow. And so it keeps the leaf litter on the steep slopes, and they actually like steep slopes. They don't like being in the flats. And that protects the soil surface from erosion. Most of the nutrients like nitrogen and phosphorus and potassium that you would be applying to your garden plants, that's in the top little layer of soil. And so having that mat on top of it keeps it from being eroded out. And that reduces the loss of nutrients from your forest ecosystem. It keeps the tree growth, growing well. And it reduces the export of sediments. And that's really important for tropical reefs, because if you get a bunch of sediments being washed out into the water, it starts shading the algae, which are endosymbionts of your corals and your other organisms, and then it also interferes with their feeding on zooplankton. And so they waste a lot of energy trying to throw that sediment off of them. So these things are really important in terms of maintaining the ecosystem. You can see the fungus on the left, and you'll notice that the leaves are bleached. And in fact, they use a chemical to bleach the leaf litter. And if it was a wood rotter, the same thing. What they're doing is they're taking out the lignin. Lignin is the stuff that makes wood hard, so that they can better get at the cellulose and the goodies that are in there. So the one on the left with the mushrooms is called a white rot. And it actually does decompose leaf litter, or if it was wood rotter, wood faster, because it's able to break that lignin. Here on the right, you don't have those. You can see the darker color. The leaves are not bound together. These decompose much more slowly by microfungi, ones that never produce a big fruit body. So one of my colleagues, when I first moved to Puerto Rico, was an aquatic biologist. And we came to the conclusion that the rainforest floor was acting like a stream, and there were debris dams out there, and these fungal mats were debris dams, keeping things in place. Normally they talk about debris dams in streams, snagging on rocks. So we had to do an experiment to actually show that these fungi were important in keeping leaf litter up on the slopes. And to do that, we picked slopes of different angles, going from about 18% up to 80, 82%. That's like this. So you're probably wondering how we did that without falling off and knocking all the leaf litter. We use ladders. And no. So we were reaching through ladders, and then we'd reposition the ladder. And so what we did was we got freshly fallen leaves, and these like fresh fallen leaves, marked them with whiteout, put a number on each one, and then a corresponding number on a little plastic stick, which was a pipette stick. And we then released them all at the tops of these slopes. And every 2 days, we would go and we would pull on them and find out if they were attached or not. So we would record that. We would measure the distance that they had moved down slope from their corresponding numbered stake. We would move the stake up, and all of the odd-numbered ones, we would break the connections and put them right back. So we had a control where we let the fungi catch them, and we had the ones with the broken strands. And if you look with the low slope, there's really no difference between the two treatments. And the greens are the broken ones. And, well, no, the greens are the controls and the pinks are the broken ones. As you get higher and higher in the slopes, you can see there's a real difference, particularly in that one on 80% slope. So they are making a difference in terms of hanging on to them. If you go on the walk with me on Saturday morning, you'll see erosion columns in places where the litter mats are absent in the botanic garden. And an erosion column has a piece of leaf litter or a little rock, and the pounding rain just erodes all the soil downhill. It just pounds it. And we'll see lots of this, which are roots that have been eroded out of the soil. I had an undergraduate intern for the summer, and she wanted to measure how much soil is actually exported from a steep slope. So she went along the driveway of our field station, which had a very steep bank, and she found litter mats, but different percentage of cover on, the ground. And those are all of those dots. And the ones that we had traps at the bottom, metal traps, to catch the sediments. And it was only 3 weeks that we had to do this experiment. Yeah, okay. And I didn't think we were going to get anything measurable, but we did. And here you can see 60% cover, 80% cover. We only lost 3 grams of dried soil off of that little 1/4 of a meter length. That's something. You get onto only down to 40 or 50% cover, and we lost 6 to 9 grams of soil off that little tiny stretch. And then all the way over here, where we only had 25% cover, we lost 30 grams. That's a lot of soil going downhill. So makes a difference. So how does climate change affect all this? You'll see the top line and the bottom line are highlighted. Climate changes have 4 different ways of affecting these litter mats and their function. First, there's the increase in rain intensity that you get because warm air holds more water, and storms tend to track more slowly. So that just ends up in more erosion. If you have drought, the mushrooms shrink, and the mats sometimes die, and you get more erosion when the rain comes back. If you have higher heat, that makes them dry out faster, the mats shrink, more erosion when the rains come. Hurricanes opening the canopy. Now that's interesting because it has 2 different ways that it affects it. It dries out the forest floor litter, particularly up on ridges that are exposed to wind and sun. And there's less leaf litter up above. Okay. There's less leaf litter falling to feed the mats and so they shrink, and you get more erosion. So I'm going to show you some examples from experiments that we've run. And this is the effect in some control plots where we haven't altered the forest canopy. And we had the El Nino drought in 2015. Now, when you do experiments, you usually do replications, and that's what the 3 different bars of the different colors are. So they're different plots in different locations in the forest, but the control and treatments are paired up. This is October, wet season, before the trim, or before the drought. And this is the October 2015 drought. Look at the drastic drop in percent fungal mat cover on the soil. Real shrinkage. Now, we ran and are still running another long-term experiment. We wanted to see what would happen if we increased the frequency of strong hurricanes above background. Now, how do you do that? We used arborists to trim the canopy to the same extent that a natural hurricane takes down the canopy, and transferred that to the ground. And then we had control treatments. And if we looked at what happened in the control treatments, there was a drastic drop in the percent mat cover in the cut versus the control. But there was one control plot in the both in the cut treatment comparison and in the drought of 2015, that one control plot did really well. Even though it was, the difficult conditions. And the answer is that first fungus that I showed you, that horsehair marasmius, there you can see the strands of the horsehair-like rhizomorphs. This is a tough fungus. It lives most of the time suspended in shrubs above the forest floor, and as soon as it stops raining, conditions get really dry. So this fungus is sort of pre-adapted to deal with drought. And in Block B, control, it had a lot of boulders. About 25% of the ground was covered in boulders. And boulders are sort of like living up in the air. When it stops raining, it's dry. So it had all the rocks making it dry. And it also had 2 tree falls before we did the measurements. So it had exposure to canopy. And so this fungus was there in droves, and it was the only one that was dominant after the drought. So I had another summer student, and she got the bright idea that we had all these steep road cuts, including the one to the driveway to our field station. And could we use some of these fungi that we knew were really super tough and put them out there on these steep slopes? And the problem with those road cuts is they get more sunlight, so more drying. And so we took 4 different species that we thought would be worth trialing. And so she found colonies of each of these. This is our horsehair marasmius. That's what it looks like when it fruits. Those are actually 2 millimeters in diameter. And it only fruits when it's been raining for 3 to 5 days constantly. Then this is one of its relatives, another marasmius, Marasmius guyanensis, also has rhizomorphs. All of these have black rhizomorphs. Micromphaly breviphys is usually on little twigs, and it has gelatin in the cap, which helps keep it moist. And then another Micromphaly that was up in the air. So we had those. So she divided each colony in half. She put one half in partial shade and the other half in the full shade. So the partial shade are the gray in front, and the full shade are the black in back. And not surprising, our super fungus, Marasmius crinisequi, which literally means hair horse, didn't change. That's not a significant difference. So it didn't blink. So it does equally well in half shade as it does in full shade. Marasmius guyanensis didn't do quite so well. It's one that lives on the forest floor, but it was still something that could be usable. Micromphaly brevipees, we were surprised it actually grew better in partial shade than in full shade. But the other one, the one that's in the same genus, that's up in the air, did not do well in partial shade. So it's sort of the Harvard rule. You control the humidity and the temperature and the light and everything, and the organism does as it damn well pleases. All right, here we go with heating. One of our colleagues in Puerto Rico, Tana Wood, set up an experiment to elevate the temperature in the rainforest 5 degrees centigrade above ambient. And what you see on the left is one of these small plots, which is the size of a trampoline. And there are fake heaters above the controls that are paired with the plots that have the real heaters that are heating. And we used our super fungus again, Marasmius crinisequi. We had a feeling it would do well. And we chose one of our wimpy ones. This is Gymnopus johnstonii, and it has these superficial white mycelial fans, which make it susceptible to drying. And on the left, you see the blue. These are the control plots. This is our wimpy Gymnopus johnstonii. This is our super fungus, Marasmius crinisequi, and there's no difference. And these are actually not means, these are medians. That's the middle, which is better for when you have very highly variable data. Now, if we look over here in the red, you see the effect of heating, and the Gymnopus, which is our wimpy one, greatly reduced, very different from that. But the Marasmius, nope, same, not different. So now we know that heating that's associated with climate change will knock back certain fungi, but there's a certain resilience in the ecosystem because some species can handle it and take the heat. So what does removing the canopy by a hurricane do? And I told you about our experiment where we trimmed the canopy to mimic hurricanes. This is a real hurricane. This is Hurricane Georges, and this is behind our field station in Sabana in Puerto Rico, and it's actually where the heating experiment is going on now. So there's a lot of exposure to light on the forest floor, and there's very little leaves up there to feed those mycelial mats. So this is me measuring the extent of these leaf litter mats made by fungi on the forest floor in the control on the left and in our trimmed plots. Big difference. And here we can see the effect on the percent mat cover on the forest floor for those two treatments. The blue is in the controls, and the reds are the trim. These are two measurements done before the trim, which is here. These are different seasons. This is the dry season, and this is the wet season, which is why this one's higher. Then the trim, and then it was dry season, but the control actually did quite well, but not the trim. Okay, and April dry season, even drier, yeah. So it does make a big difference. Now, my favorite fungi. Ooh, now you can see why I like them. They come in day-glow colors, usually. Yeah. Most of these are species that are undescribed, except for these 3 on the lower left bottom. But in fact, the 3 on the lower left bottom, we have since discovered with DNA sequencing that they may look alike on different islands, but they're not the same. So we have what's called cryptic species. Now the other ones are ones that are in the process of being described. And I have to come up with names for one. This one is from Belize. I'm giving it a name which in Latin means long red legs. This one is yellow on top, and you turn it over, and it's brilliant red underneath. And it was so shocking to us that we almost fell off the ridge and the highest peak in Belize. And so we named this Mirabilis, which means surprise. So this fantastic diversity, and what's amazing about these is that you get these related species on different islands and in the mainland, but they're not the same. So this is just one lineage. These are the purple and green ones. And they're all related to each other genetically. This sort of looks like a family tree, but it's not exactly the same as a family tree, because in a family you would know who died. In evolution, particularly with things like fungi that don't leave fossils, you don't know who's missing. And yeah. And this one, which is the most basal, is probably a very old origin species. So it's down there in the base, but it may not have been the one that gave rise to all those others. It's just down there, very old. And if you'll notice, it says Guyana. That's the Guyana Shield. That's one of the very oldest pieces of terrain there is. And the Guyana Shield and the Amazon Basin are considered museums of ancient species, but they're also cradles of evolution and new species. So it does both. But if we look at some of these, this is one of the green ones that is from Belize, and it's related to the green one in Puerto Rico. And this one I'm naming Little Emerald. But this one in Puerto Rico, we've now found has a sister species in the Dominican Republic. And they have some similarities. They have a little white skirt around the edge. But this one has a hole in the middle and that one doesn't. And that one has more purple on the cap and this one has less purple. And that one's got a skinny stalk and this one's got a thick stalk. And the genetics says they're no longer the same species. They're sisters. And we get the same thing for this pair up here. This is a species I described about 25 years ago from Puerto Rico. It's purple with green gills, and it's related to this one from Guyana. And again, they both have a little white skirt on the edge, and they have green gills and a purple cap. But they look pretty different, don't they? Yeah. And then down here, we have this one that's related, but it's much more basal, and that's in Belize. And it's a tall, skinny thing. But again, the same color scheme. And then we have this one, Hygrocebi appalachiensis. That's from the southern Appalachians. And it turns out that the southern Appalachians were a refugium during the glaciations, and so it retained things that otherwise went extinct.
Can I ask a quick question?
Yeah.
What makes them green? Is it chlorophyll?
No, it's not chlorophyll. Some of the colors are. I'll have to come up with a name. There are specific chemical colors, and they can actually change color depending on an oxidation state of the chemical. So purple and green or red and green are not necessarily different from each other. Yeah. Okay, now we get into sex. And so why do you get all these things that are related on different islands, but they're not the same species? And how do they get around? So fungi in general, particularly these mushroom fungi, have a lot of compatibility factors that determine whether two hyphae that came from different spores can mate. And a lot of mushrooms, including the wax caps that I look at, have not one place on the chromosome that determines sex. They have places on two different chromosomes that determine sex. And so they're not called male and female, they're called big A and big B and little a and little b. And so you don't need a menage a trois to have sex. You just have to have exact opposite mating types. And when they decide that they're compatible, they fuse. And in most fungi, mushroom fungi, they don't fuse the nuclei. So they're not like us where at fertilization, the egg and the sperm fuse, which would make it 2N. These keep their nuclei separate, and they run around in pairs like they were holding hands. And it's called N plus N, or dikaryon. And so here's the happy fungus. And when one goes to make a new segment, each of these divides mitotically. They make identical nuclei, and one pair goes off in the new segment going through this little side tube, and the other two stay behind. They don't get together and fuse until they're up in the mushroom cap, right before the spore production, and then they fuse, they go through meiosis, out pops all four sexes, okay? Well, that's the standard. Okay, so with that background, I'm going to go through a thought experiment. Imagine that you're a spore flying from another island, say Martinique, and you're landing on Saba. These are all restricted, except for one species, to moist or wet forests. So they have to land somewhere wet, like the donut around the top of Mount Scenery, or a little above or a little below, okay? Now, we also know, even though we can't grow them, we have enough information to know that these are symbionts of plant roots. We don't know what they're doing in there. We don't know if they're beneficial or detrimental or neutral. And we don't know whether they're host-specific. We know one that's host-specific, but it's not a wax cap. But, okay, maybe it has to land within 1 meter of a special host. Maybe it can take several different hosts. We don't know. But it does have to land within 1 meter of its exact opposite mating type. And it has to do that within like a year or a couple years in order to be able to mate. The probabilities that we're looking at are like a snowball's chance in hell. Okay? So how do they do this? Well, this particular group has something that's different. It only occurs in a couple of the other wax cap lineages. And that is, it makes big basidia, which produce big spores, and it makes little basidia that make little spores. And the big spores, you see all these circles, that's oil. They're packed with goodies. And we know that those spores have more than one nucleus in them. We don't know whether they're opposite mating types or whether they're all the same, but based on the phylogeography, I'm betting that at least some of them have opposite mating types all packed up in the big spores that's very well provisioned. And it's those that make it to another island and can then make a reproducing fruiting body and generate more of itself. But there's more to it than that. Let's do a little side thing. You might think that dodging sex is unusual, but in fact it's not. We heard parthenogenesis described in corals yesterday, and that's where they're asexually reproducing. In plants, it's often called apomixis. It can either be that they're putting identical nuclei into a seed coat, even though it hasn't gone through any sort of sex, or that they're selfing. And some plants will actually stagger the development of the anthers with the pollen after the stigma. And if the stigma goes for a day without being pollinated by another plant from far away, then the pollen develops on the anthers and it self-pollinates. Yeah. The plant pathogen on cacao also is selfing. I'll show that a little later. Some mushrooms actually produce fruiting bodies that only have one mating type, so they look like they're a sexual fruit body, but they're not. Other fungi put nuclei of opposite mating types together in the same spore, and I think that's what the wax caps do. But as I said, that's not the end of the story. You have to then have something that's called genetic drift. So you've had a spore with opposite mating types land on the top of a mountain or partway down. And through generation after generation after generation, it sort of drifts off in one direction. And then another spore makes it from the same source and lands. And they say, oh, I don't like you. You're too different. I'm not going to fuse. And so you get them going their separate ways, and they're incompatible. And so you can actually get multiple species of these green and purple ones on the same island, like Puerto Rico and the Dominican Republic, from this staggered migration and drift. Oh, now Halloween stories. This is the jack-o'-lantern fungus. This is pictured in southern Appalachians. And the first thing that anybody asks me is, why do they glow in the dark? And there are two different hypotheses that compete. They may actually both be correct, but in different proportions. The jack-o'-lantern fungus is very poisonous, deadly poisonous. This may be a warning to night foraging fungivorous animals. Do not eat me. Okay? This is the other hypothesis, and that is that one of my colleagues who's worked in Hawaii found that this particular group of mycena, when they're babies, you can see the hairy one up top in the button stage, they have those sticky sort of projectiles sticking out of them. And those get picked up by snails and collembola and other invertebrates that are running around, eating fungi, and they get stuck to their back and they get carried somewhere else, and every one of those little tiny bits can make a new mushroom. Okay? And the one down below is not the same species as the one on the upper left, but it was pictured in regular light and also under just dark. Yeah. Sugared. So the sac. And you can see all these tiny little things all over it. And same here.
It's like frosted flakes.
It's like frosted flakes. Except they're sticky. All right. Okay, so that was a good hypothesis, except that these are all bioluminescent mycena that we described from Brazil and Puerto Rico. And none of them have those sticky little projections on them. So there goes one hypothesis down the drain. I think it probably works for the sugar caps. Doesn't work for most of the mycenas. Okay. Now, do we know anything about toxicity in these? No, we don't know about toxicity in the bioluminescent ones, but we do know about this one. This belongs in the Mycena pura group, and they all have purple colors and they smell like radishes. And a group that works on developing drugs in Denmark or the Netherlands check this thing out to see if they can make something useful drug-wise out of it, and found out that it stops all cellular processes. Now, if you think about that for a minute, that's equivalent to it kills you. Okay, we have a saying in mycology that there are old mushroom eaters and there are bold mushroom eaters. There are no old, bold mushroom eaters. Okay, but this is a bioluminescent fungus that people eat. This is the honey mushroom, and two of these species are called the humongous fungus. Okay, and there's one out in Michigan, which was the first one. That's the one in the upper right. And then the one out in Oregon, which they found later. So the Michigan one is only 2,500 years old and 6/100ths of a square mile and only 400,000 kilograms. While the one out in Oregon is more than 8,000 years old, 3.7 square miles, and more than 3 million kilograms. So Oregon won on the humongous fungus.
What do you mean by the square miles? That's where you find it?
That is where one individual is found. The extent of one individual.
Very expensive restaurant to eat.
Okay.
That's the cells of the mycelium, right?
Yeah. So we're going to get into why is it all one individual in a minute. That is rhizomorphs. This is also called the shoestring fungus. It's also called foxfire because it makes logs that it's just decomposing glow in the dark. Okay. So the question is, how does it get so big? That's what everybody misses. So that's what we're going to look at in two different things. One thing is that dead trees provide the energy for it to produce toxins, which it pumps out through those root-like structures, which is this is also called the shoestring fungus. And those pump out toxins at the next row of trees and kills them, and then it eats them. So it's not a parasite. It acts more like a predator that kills and then eats and then uses that to kill the next ones. All right? So this is what one of those kill patches looks like. And they have it diagrammed there. These things can be hundreds of acres. Yeah. So the other thing of how did it get so big? And now we get into the invasion of the body snatchers. And there's very little in science fiction that wasn't already invented by fungi. So we're back at sex. This particular fungus actually does fuse its nuclei and becomes 2N, diploid. But there are other ones that are N plus N. Either way, you can get a single spore making a mycelium, and this fungus is growing out bigger and bigger, and it runs into it, and the single spore one says, I see genetic material that is the opposite mating type from me. And the humongous fungus plays femme fatale and says, hi there, I'm compatible, come fuse with me. And they do, and this one displaces, which is a nice word for saying it kills off all of these nuclei and takes over its body. And that's how, when they did the genotyping all the way around that huge 3.6 kilometer ring, all of it was the same. If you're getting these diamond crosses where they'd fuse, they would normally have a sector that would radiate out, and then you'd quickly have a patchwork of half siblings. But that's not the humongous fungus. It's all one genotype. So now the Dr. Jekyll and Mr. Hyde fungi. This I watched for years, and it looked like a fairly innocent fungus, just growing on the surface of leaves of a shrub. And then the next week I went out, and that's what the very same leaf looked like. Oh. And if you look where the white arrows are, you can see the bits of green still there. And so what this fungus is doing is it's changing from being what's called an endophyte, which is a fungus that lives inside of plants without causing damage. And then suddenly something triggers it, and it produces toxins and kills its host and eats it. So this is an undescribed species of mushroom. It is related distantly to our horsehair marasmius. Our horsehair marasmius is also one of the Dr. Jekyll, Mr. Hyde. It turns out to be a suite of species, but they all act pretty much the same around the world in the tropics. And this one, they know what it is that it's using to make the leaves dead, like that. It uses ethylene. Ethylene is what they use to make apples ripen, that they've been held in the, And so this is saying, okay, time to senesce, bam. It's already got a hold of them because it's already attached to all the leaves. So Dr. Jekyll, Mr. Hyde. And then this is a weird one. This is a fungus that was discovered in Guyana, neighboring countries, killing citrus, but it also kills other trees and shrubs. Now you look at that and you see all those little knobs and you think, oh, that thing's producing spores. It's been studied for 45 years. Nobody has ever seen any sort of spore structure on these things. What the heck's it doing? No spores, no problem, take a lift. Okay? It's getting a helping beak from a feathered friend here to incorporate it into its nest. And they're moving it around. Same thing for horsehair marasmius. And then I promised you that you would see the sulfing mushroom. This is the one that attacks and kills cacao, which is where we get our chocolate cocoa from. And again, it lives like an innocent endophyte inside the cocoa tree as long as the canopy is closed. And then something opens the canopy like a tree fall, or the farmer wants his cacao to grow faster or less rot on the pods, opens it up. The end starts growing very quickly. The fungus says, yeah! And it quickly selfs so that it can produce those fruiting bodies. And it turns into a pathogen and kills the fast-growing shoot, which becomes the witch's broom, and then it fruits on that and spreads then to new trees. Now we have the wood decay fungi that kill fungivorous nematodes for their nitrogen. So wood is a pretty tough place to live because there's not much nitrogen, but there's lots of carbon. And everybody needs nitrogen to make proteins and enzymes and all kinds of good things. So everybody's out there trying to hustle nitrogen. And so these fungi are turning the tables and eating the nematodes that are out there eating microorganisms. So this one on the far left is called Pluteus. This one's a shaggy mane. It's a type of Caprinus. They share the same kind of trap. They have hooks, like grappling hooks on their hyphae, and they snag the nematode, and then they grow into them and eat them. This one on the right uses deadly sweet trick-or-treats. It makes little lollipop-shaped knobs on its mycelium that are sweet, but they're toxic. And it paralyzes and kills the nematode that puts its mouth on them and then grows down the gut and eats them. Okay. I'll describe two others, but if you want to see more about these, there's a YouTube video made by SciShow, and it's called The Carnivorous Fungi That Devour Nematodes. And this is really worth watching. I had my husband watch it, and he got scared. There's... No. So there's the constricting ring trap, which is actually from a mushroom. And it has 3 cells. And they have a motion sensor or touch sensor on them. And so if a nematode puts its neck through that ring, the cells on that triangle suddenly swell, they lasso it, and then they grow in and eat it. Okay. Then the one that really got him was the one that fires harpoons to trap their nematodes and eats them. So you got to see this, particularly for Halloween. And then the zombie fungi, okay? The two on the left, that's a former beetle grub. That's a former grasshopper. They don't really change the behavior of the animal. They just die in place, and then others of the same species encounter the spores. But these two on the right, the jumping spider and the ant, the fungus actually controls the mind and controls the behavior before death so that it puts it in a very nice place to disperse spores to others of its same species. So they usually have them go up somewhere high and then fasten them to a surface, or in the case of the ants, it makes them attracted to light. They crawl up a stem or a leaf, and then it makes their mandibles clamp down on it so that even after death it's still hanging up there and dispersing its spores. Okay. And then this is a cicada. We've had a big cicada outbreak in the eastern US this year. We've seen lots and lots of these. Matt Casson in West Virginia University calls these the salt shakers of death. It completely replaces the genitalia of the cicada with spore masses of the fungus parasite. And as the cicada is flying, all these spores are being spread, like like a plane duster, a crop duster. So, and then this is fungi that are vampires on plants by proxy. This fungus is actually parasitizing or killing scale insects, which are then feeding on the stems. But what they make sure is that they leave the proboscis of the scale insect in place, which is sticking into the phloem, which is where the sugar is being translocated in the plant. And so they are sucking up the sugary sap through the live or dead scale insect. And at that point, I will take questions. One in the back.
In your studies with drought at the beginning of the presentation, how confident are you that the fungi, given their rapid rate of growth, are not mutating and adapting to the drought or the moisture conditions rather than just being themselves?
There's a certain minimum amount of okay, I've got to repeat you. So how do we know on the fungi that responded to drought whether they're actually evolving the capacity to adapt to drought or not? It takes time for evolution to happen. So it would take probably at least 10, 20 cycles. Yeah. The fastest evolution I've heard is with a fungus in the Netherlands, and that was pretty quick. Some of the fastest evolution actually happens with hybridization between species. So even though there's a lot of incompatibility factors that prevent interbreeding, it occasionally happens. And the furthest distance apart genetically that we've seen that be successful is 3 to 6% difference. You get beyond that and you get things that can't reproduce. They're dead ends, and we have found some. Yeah. Uh-huh.
The experiment that you did with the heat sources,
Mm-hmm.
did you measure humidity levels and look at, I'm just assuming that
Mm-hmm.
when you use the heat lamps or whatever, the humidity is going to be reduced.
Mm-hmm. Okay, the question is whether the humidity was measured in the heating experiment. And yes, the person who's running it does measure humidity and was also measuring moisture with moisture meters. However, the experiment that we did with partial sun and full shade, we did a simple way of measuring humidity. And it wasn't your normal way, with a humidity meter. What we did was we took little tubes with water in them and a wick, and we placed those where the fungal mats were, the ones in the half sun and the ones in the shade, and then measured how fast the disappearance of water from the microtube because the lower the relative humidity, the faster the wick draws water out of the tube, and so it's evaporation potential we are measuring, and that was highly significant between the partial shade and the full shade. Another question?
For the graph that you showed of the experiment that you were just talking about with partially shaded and full shaded, is there any kind of internal processes that could require direct sunlight?
Okay, the question is, are there any processes in the fungi that would require direct sunlight?
Because you said there was one where it actually did better in half shaded
Yeah.
than full shaded.
It is a possibility. For one thing, in the experiment that the student did with the heating experiment, he found that if it was in the rainy season when there was plenty of moisture, the wimpy fungus and I think also the marasmius grew faster. So the heat was speeding it up as long as there wasn't a moisture deficit. Yeah.
So probably more the heat than the sunshine.
Yeah, it's more the drying. There is something, though, in the wax caps that's quite interesting, and that is that if you get these wax caps that are covered in leaf litter, before they've pushed their way through, they're oftentimes very pale colored or completely white. And the betulin pigments, which are most of those really bright colors, betulin is the type that's in beets, which is where it gets its name from. If it doesn't have the sunlight, it doesn't develop the pigment into a visible form. Uh-huh. Yeah.
Thank you, Jean. That was a lot of awesome information to remind us to appreciate all the fungi we see and to be scared of some as well. For those of you who arrived a little late, know that she is doing her official field project Saturday morning. You can sign up with us. We're optimists, so we're just keeping our schedule as it is with the movie being changed. And we want to also make a shout out to the Sunny Valley Youth Center, Elka Charles and her family, for letting us use this facility tonight. Of course, it didn't rain, it sounds like. Maybe it did. And for those of you at home, know that Island Flavor is our official sponsor this evening. Fantastic place for dinner. And if you're not with us, sucks for you because lots of people are heading over for a great dinner, focusing on mushrooms. And we will see you at Amanhana on Saturday night. Thank you very much. Whoops. Yes.
We're only doing like 20 meters of the beginning of the Mount Scenery Trail because I know where the fungi are. Okay. That I want to show you. But then we're going through the botanical garden. And after that, we're ending up at Colibri Cafe. And I'm going to do a demonstration on how to use iNaturalist. So if you want to learn how to use iNaturalist, if you don't know how to use it and you have a cell phone, I have a sheet on how you get it loaded into your phone and how you get your profile set up. I can give you one of those, but we can use the Wi-Fi there at Colibri, have something nice to drink, and do a little demo on iNaturalist. It's really wonderful because not just fungi, plants, anything. You can put in a picture and you don't know what it is. If you can at least say plant, fungus, animal, invertebrate, insect, you'll get the AI doing a lot of the identification, and it'll immediately come back with a bunch of choices, and you can look at the choices and say, oh yeah, it's that one. But you can also get all these experts, which a lot of them are not PhDs. They're amateurs that are so good. We call them paraprofessionals. And there are networks of paraprofessionals, including one of my colleagues over in Puerto Rico. So he is wired, and he'll be responding, and I'll show you how to send messages through iNaturalist to get help on IDs. Okay?
Thank you. Great.